Welcome to this video on the early development of C. elegans, a premier model for developmental biology and invariant cell lineage. This video is divided into two parts: early events and specification and gastrulation. We will cover topics from fertilization and symmetry breaking to autonomous and conditional cell fate specification, culminating in a lineage summary. C. elegans development features 959 somatic cells formed through a rigid, predictable pattern. A single decisive event, the entry of the sperm, breaks the symmetry of the apolar oocyte and establishes the embryo's axes. The sperm entry site is crucial, becoming the posterior pole. The sperm donates a centrosome, which organizes local microtubules, initiating a cytoplasmic flow essential for cell polarity determination. Microtubule reorganization causes asymmetric distribution of PAR proteins. PAR3, PAR6, and PKC3 are restricted to the anterior cortex. Conversely, PAR1 and PAR2 accumulate at the posterior cortex. P granules are swept posteriorly to germline precursor cells. Cleavage is rotational holoblastic. The first asymmetrical cleavage of the P0 zygote produces a large anterior AB cell, which is a founder cell, and a smaller posterior P1 cell, which retains stem cell characteristics. During the second cleavage, the AB cell divides meridionally into ABA and ABP. The P1 cell divides equatorially into the EMS and P2 cells. The dorsal ventral axis is established as the ABP cell is squeezed dorsally against the eggshell, while the EMS cell remains ventral. This image illustrates the orientation and products of the second cleavage, marking the transition to the 4-cell and 8-cell stages. Autonomous cell fate is driven by internal factors. The SKN1 transcription factor is essential for the EMS cell to produce the mesoderm and endoderm lineages. The PIE1 protein autonomously protects the P lineage, preserving its germ cell fate. Conditional cell fate depends on cell-cell signaling by the P2 cell. P2 signals to EMS via MOM2, polarizing it into gut and muscle precursors. P2 also signals to ABP via APX1 and GLP1, distinguishing the dorsal ABP from the ventral ABA cell. Gastrulation begins remarkably early, at the 26-cell stage. It is initiated by the inward movement of the two E cells, the gut precursors, creating a small blastopore for subsequent cell entry. Following the E cells, P4, MS, and D cells enter the blastopore in a strict sequence. Epiboly, the expansion of the AB descendants, the ectoderm, covers the embryo's surface and closes the blastopore at the ventral midline. The lineage summary table provides an overview of the major derivatives from the 6 founder cells. The AB founder forms the nervous system and hypodermis, MS forms muscle and pharynx, E forms the gut, C forms muscle and hypodermis, D forms body wall muscle, and P4 forms the germline. Key exam facts include: sperm entry triggers symmetry breaking. The P2 to EMS signal is driven by the WNT pathway, MOM2. The P2 to ABP signal is driven by the delta Notch pathway, APX1, GLP1. PI1 is crucial for preserving germline fate. Mastering C. elegans lineage is fundamental to understanding developmental biology. Thank you for watching this video. The images used in this video were sourced from the websites displayed on this slide.